Hello everyone, welcome to this video on the principles of endocrinology, so all about hormones and the endocrine system. I'm going to be taking you through this system in 5 parts. So firstly we're going to look at the principles of endocrinology, then we're going to have a look at some examples of glands and systems. So we're going to look at the adrenal gland, thyroid hormones, growth hormones and energy balance and how the hormone system feeds into that. So firstly, principles of endocrinology. So hormones, what are they, why do we need them, and where are they formed? So hormone, the definition is a substance formed by a specialized gland in small amounts, carried in the bloodstream to a target organ where it has a specific physiological effect, and it's required for homeostasis and communication in the body. So we'll have a look at where hormones are secreted from later. 3 modes of hormone action are number 1, development, homeostasis, and number 3, adaptation. So we've got development of physical, sexual, and mental characteristics, for example, in the embryo, and in homeostasis, feedback, for example, blood glucose, and in adaptation, adjustment with respect to circumstance, for example, fight or flight, which we will have a look at later. So firstly, we're going to have a look at the 4 classes of hormones. So a hormone can be a trophic hormone, which means that it stimulates the release of another hormone, or an effector hormone, which has an actual effect on a target organ. So we have peptide or protein hormones. So these are just proteins made of amino acids. They can travel in the blood. Then we have steroid hormones. So these are cholesterol-derived lipid hormones. They're lipophilic and they're not soluble in water, so they need to be bound to plasma proteins to travel in the blood, and they also need to be released from these proteins to have their effect. They can't be attached to them. Amine hormones, so some examples are dopamine, which makes epinephrine or adrenaline, and T4, thyroxine, serotonin. So amine hormones are derived from the amino acids tyrosine and tryptophan, and they have that NH2 group. Arachidonic acid derivatives, so these are derived from linoleic acid. They are pretty important in inflammatory response, as you can see by these examples, and NSAIDs or non-steroidal, so like aspirin type medicines, can inhibit the production of these hormones by inhibiting this enzyme called cyclooxygenase. So some examples are leukotrienes, prostaglandin, the prostacyclins, thromboxanes, and prostaglandins, so all important in the immune system. Let's have a look at peptide hormone synthesis and processing. So this was the first class of our hormones. So the processing happens co-translationally. So a peptide hormone with an N-terminal signal sequence is transcribed and then translated by the ribosomes, and this N-terminal signal sequence binds a molecule called a signal recognition peptide, which directs a protein to a secretory pathway. The SRP peptide is a 7S RNA peptide, and it brings this peptide hormone to a secretory pathway, which causes translational arrest as well. So translation stops. The SRP docks on the endoplasmic reticulum membrane. Translation starts again, and the signal sequence, so the signal sequence is cleaved off the peptide in the ER. It might require additional cleavage, so here we have a pre-hormone with a signal sequence, and that needs to be cleaved off to leave the normal hormone. And here we have a pre-pro hormone, which has an additional pro sequence, and these both need to be cleaved off. Amide group addition is also an important modification for some hormones, such as the RF amide family. So these are added at the C-terminus. And 2 examples are kisspeptin and GnIH, gonadotropin inhibiting hormone. So these are all related to the sex hormones. And kisspeptin is an arcuate and antroventral release from the arcuate and antroventral periventricular region of the hypothalamus. It increases gonadotropin releasing hormone secretion from the pituitary, so increases the production of LH and FSH. Gonadotropin inhibiting hormone is released from the paraventricular and dorsomedial region of the hypothalamus, and it decreases the release of gonadotropic hormones. We have 4 major hormone signaling mechanisms. So we have tyrosine kinase activation, G protein adenyl cyclase pathway, DAG IP3 pathway, and a cytoplasmic or nuclear receptor pathway for steroid hormones. So we'll go through these. Number 1 is tyrosine kinase activation. So this is all about how does the hormone cause a response and the molecular detail of it. So we have binding, dimerization, phosphorylation, and response. So number 1, the hormone binds to a tyrosine kinase receptor. For example, insulin or insulin-like growth factor. These are all tyrosine kinase activated. Now, the tyrosine kinase activates, so 2 of these come together, they dimerize, and that activates the tyrosine kinase activity, which is intracellular. So number 3, the tyrose kinase activity phosphorylates various molecules inside the cell to cause a cascade of phosphorylation using ATP and causes a response. So number 2, we have the GS or GI adenyl cyclase. So we have binding, G protein activation, adenyl cyclase activation, CAMP production, which causes activation of PKA, protein kinase A, and then phosphorylation cascades. So the hormone binds to a GPCR, G protein receptor. Then this G protein receptor is coupled to a G protein, which uses GTP to is phosphorylated by GTP. So GS stimulates adenyl cyclase, whereas GI inhibits adenyl cyclase. And then, depending on which one has been stimulated, you get more CAMP formed from ATP or less CAMP formed from ATP. CAMP, the action is to activate PKA, protein kinase A, which then causes phosphorylation cascade. So PKA phosphorylates CREB, for example, and then that causes the transcription of genes with CRE activation domain, which causes a response. And it can also phosphorylate serine or threonine amino acid residues on various proteins, which also causes a response. So that's the G protein cascade. Now we look at the DAG or IP3 pathway. So this is the diacylglycerol, or I can't remember what this stands for, to be honest. So oxytocin and gonadotrophin releasing hormone, these are examples of hormones which signal via DAG IP3 pathway. Similar to the GPCR, the last pathway we looked at. So the hormone binds to a GPCR, and then this time it activates GQ, another G protein which gets phosphorylated, and then GQ activates PLC. So PLC is an enzyme which converts the molecule PIP2 into IP3 and diacylglycerol. Those two then go on to perform their various functions. So IP3 causes calcium release from the endoplasmic reticulum, which causes calmodulin activated protein kinase to be activated, which causes phosphorylation and a response. And then DAG activates protein kinase C, which causes phosphorylation and then a response like that. Now, cytoplasmic or nuclear receptors. So these are receptors which are inside the cytosol, and they're pretty much signaling for every steroid hormone or lipid hormone, because these can enter the plasma membrane. So this is the primary structure. So we have an AF1 transactivation domain, a DNA binding domain with 2 zincs, zinc finger portions. Then we have a ligand binding transactivation, dimerization, and repression domain with AF2 transactivation domain here as well. And then this folds into a tertiary structure that looks a bit like this. So this is AF1, zinc fingers, the ligand binding area, and you can see a ligand has binded here. So the plasma membrane, we have the ligands that we want, steroid hormones, which can pass in through the membrane and bind to this intracellular receptor. The intracellular receptor is usually connected to an HSP in a complex, and then once the ligand binds, it dissociates and moves into the nucleus. It dimerizes and attaches to a response element, which can activate a gene and cause target gene transcription. So those are the signaling mechanisms. Now let's have a look at the hypothalamus and pituitary, which are very important for the control of other glands. So this is the structure of the hypothalamus, and this is the pituitary down here. We have these many nuclei in the hypothalamus, but these are the most important ones. So we have this one, which goes to the anterior pituitary. Then we have the paraventricular nucleus and the supraoptic nucleus, which give neurons directly into the neuron hypophysis or the posterior pituitary. Now, the posterior pituitary releases oxytocin and vasopressin, or ADH. Oxytocin is important for sexual development and pregnancy and things like that, and vasopressin is a kidney hormone which regulates water uptake. So the nerve endings release these hormones directly into the bloodstream, whereas in the anterior pituitary, the neurons are connected to a primary capillary plexus, which then connect to the secretory cells via hypophyseal portal vessels, and the secretory cells secrete these hormones. So these are ACTH, TSH, GH, LH, FSH, and prolactin, these releasing hormones. So there's direct neural connection from the hypothalamus to the posterior pituitary, but not from the hypothalamus to the anterior pituitary. The connection is via these capillaries instead. So let's have a look at how the hypothalamus influences the anterior pituitary secretion. So we have corticotropin releasing hormone, CRH, which is a 41 amino acid hormone, which is released from the hypothalamus and to the pituitary and stimulates the release of ACTH, adenocorticotropic releasing, it's just adenocorticotropic hormone, which is a 39 amino acid hormone. Now, TRH in the hypothalamus is a 3 amino acid hormone, thyrotropin releasing hormone, which is very small. It stimulates the release of TSH and prolactin. So TSH is a 2 subunit glycoprotein, and prolactin is a 199 amino acid hormone. GnRH, gonadotropin releasing hormone, is a 10 amino acid. It stimulates the release of LH and FSH, so these are gonadotropic hormones. Growth hormone releasing hormone, GHRH, is a 44 amino acid hormone, which stimulates, unsurprisingly, the release of growth hormone from the anterior pituitary, 191 amino acids. Somatostatin is an inhibitory releasing hormone from the hypothalamus. It's 14 amino acids long, and it inhibits the release of growth hormone from the anterior pituitary, TSH, and prolactin. These in brackets are not anterior pituitary hormones, so VIP, glucagon, and insulin, but it decreases the release of those as well. And dopamine is a monoamine, so it's an amine hormone, and it decreases the release of prolactin from the anterior pituitary. So how do we know that the anterior pituitary hormones are controlled by releasing hormones from the hypothalamus? So there's 5 important pieces of evidence. Lesions in the hypothalamus or anterior pituitary removal means that some glands atrophy. Making lesions in the hypothalamus, disconnecting the hypothalamus from some glands has the same effect as disconnecting the anterior pituitary, removing the anterior pituitary from some glands. Electric stimulation of the hypothalamus causes anterior pituitary secretions. That's pretty obvious. And then pituitary stalk transection means that some glands atrophy. Transplanting the anterior pituitary but with the hypothalamus, transplanting it by itself has no endocrine function, but with the hypothalamus together connected, there is endocrine function and secretion from the downstream glands. Addition of hypothalamic releasing hormone to the pituitary explants in culture causes function, which means anterior pituitary secretion. So some hormones have rhythms. These are the common ones. So circadian rhythm is released every 24 hours. For example, cortisol. It's highest in the morning and then drops during the day. Pulsatile release is 1 or every 2 hours, which can be things like gonadotropic hormones, for example. Sex hormones are released according to breeding season and estrous cycle. So feedback mechanisms are very important in the control of hormones and their regulation. So let's have a look at some. So endocrine tissue affects a target tissue via a hormone, and the target tissue can produce a response molecule which decreases the endocrine tissue function, so decreases the hormone, and it regulates itself so it doesn't get too stimulated. This is negative feedback. And positive feedback is when the endocrine tissue releases a hormone to the target tissue, but the response molecule from the target tissue, as it's activated, actually increases the activity of the endocrine tissue, and then increases its own function, and so on and so on. So it gets more and more until there's no more hormone to be released. So this is a positive feedback. Now you can have a look at the HPA axis, or the hypothalamic pituitary axis. So higher centers in the brain can increase or decrease the function of the hypothalamus, which secretes releasing hormones, as we just saw, to the pituitary. Now, the pituitary releases trophic hormones to a target gland, for example, the adrenal glands or the thyroid, which we will see later, and then that releases another hormone. So a short feedback loop is when a trophic hormone affects the hypothalamus. So it can either be positive or negative feedback, but the trophic hormone has an effect on the hypothalamus. And then a long feedback loop is when the target gland makes a target gland hormone. Indirect long loop is to the hypothalamus, and a direct loop is to the pituitary. So when the target gland affects the hypothalamus or pituitary glands. So last thing we need to look at here is hormone concentration measurements. We often measure these using a immunoassay with an antibody attaching to an antigen on a hormone. So you have a sample and you want to know how much of a particular hormone is in that sample, or the concentration in that sample. So firstly, in a general ELISA, which is enzyme-linked immunosorbent assay, you have your sample, and then you bind your sample to a well containing fixed antibodies, which will bind the hormone. And then you have all of this mess here. You need to wash off the unbound material and leave just the hormone. So all of your hormone in the sample should be bound to the wells. Now we add a second antibody that binds a hormone with the enzyme. So this second antibody also binds this hormone as the first one did, but it has an enzyme attached to it. And then now we can add a substrate, and the enzyme converts the substrate into a coloured product. So the more of the second antibodies there are, the more coloured product there will be, and the darker the colour it will be, so we can read that. And the colour intensity produced by the substrate will be proportional to the concentration of hormone. So now we have a competitive ELISA. So there are some cases in which we can't use this because the hormone might not have a second binding site, or it might not be able to bind. You might not be able to find an antibody that binds this hormone, another one. So what we can do here is we have a sample and a conjugate. So the conjugate is the hormone that you're looking for, but a known amount and attached to an enzyme as well. So we have the sample and conjugate. Now we bind and incubate the sample and the conjugate mixture along with everything else, and there will be competition. So the more sample there is, the more that will bind, but the less sample there is, the less that will bind, and the more conjugate with enzymes will bind. So then we wash it and we add a substrate and read. So the colour intensity here will be inversely proportional to the amount of hormone, because the conjugate, so the more conjugate is bound, the darker the colour will be, but that means that the less hormone you had in your original sample. So that's the general principles done. Now let's have a look at the adrenal gland and how that works. So the anatomy and histology of the adrenals. So you should know that the adrenal glands are located above the kidneys. They're commonly known as the kidney caps. And this is sort of a layout of the histology of them. So they're sort of triangular shaped, and these are the layers from superficial to deep. So we have the cortex, which is all of this, the capsule, and then the cortex, which is all of this here, and then the medulla. So capsule on top. The cortex contains these 3 layers called the zona glomerulosa, the zona fasciculata, and the zona reticularis. So the zona glomerulosa secretes mineralocorticoids such as aldosterone, which is involved in blood pressure homeostasis. Zona fasciculata, and then in the middle it's the thickest layer, and fasciculata is responsible for glucocorticoids secretion, like cortisol, which controls blood glucose levels and metabolism and other things like that. So it contains cells with lipid droplets containing, because these are steroid hormones, so it requires lipid to be made. Zona reticularis is the deepest layer of the cortex, and it has storage granules with cholinergic nerve supply inside its cells, and the storage granules contain the androgens, so the sex hormones that it releases. The medulla is responsible for releasing catecholamines such as adrenaline and noradrenaline, and these are amine hormones, as you might remember. So adrenal hormone synthesis, the catecholamines are synthesized from tyrosine amino acid, whereas there's a bit more of a complicated pathway to the synthesis of the steroid hormone. So we have acetate, which forms cholesterol, so these are all cholesterol derived, which then forms pregnenolone, and then this forms progesterone, which is a hormone in itself, and dihydroepiandrosterone, which then forms androstenedione, and then that forms the androgens testosterone and estrogen. Progesterone then forms 11-deoxycorticosterone, and that gives rise to corticosterone, which is the primary glucocorticoid in animals, which gives rise to cortisol, which is the primary glucocorticoid in humans, and also aldosterone, which is a mineralocorticoid. So regulation of adrenal secretions. So this is involving the hypothalamus pituitary axis again. The hypothalamus sends a releasing hormone to the pituitary, anterior pituitary, which then affects the adrenal glands. So the hypothalamus can be stimulated by stress interleukins from an immune response or circadian rhythm. These are all things that affect adrenal secretions. And then it releases corticotropin releasing hormone into the portal blood vessels. Remember that the hypothalamus is connected to the pituitary via portal blood vessels, so it releases CRH, and that stimulates the anterior pituitary corticotrophs to release ACTH, adrenocorticotropic hormone, which is potentiated by interleukins, and ACTH can also cause pigmentation, which we'll go on to later. So ACTH stimulates the adrenal glands, the zona fasciculata and zona reticularis only, so not the zona glomerulosa. And the adrenal glands produce glucocorticoids such as cortisol, mineralocorticoids such as aldosterone and androgens, so only these are affected by ACTH. Glucocorticoids, the effects are increasing vascular tone, it's increasing anti-inflammatory mechanisms, so decreasing inflammation, increasing amino acid mobilization in muscle, lipolysis in fat, gluconeogenesis, and glycogen storage in the liver. The mineralocorticoids are essential for life because they regulate the renin-angiotensin system, which stimulates sodium reabsorption and K+ excretion. So we're swapping sodium for potassium in the kidney. Androgens are the sex hormones. So negative feedback mechanism here is that the adrenal gland, the target organ, produces glucocorticoid, this hormone, which has a long indirect feedback loop to the hypothalamus, negatively regulates that, and a long direct feedback loop to the pituitary, which is a direct feedback loop. So if you have primary adrenal insufficiency, then your ACTH increases, you might get some pigmentation, and that means that your negative feedback increases, so your glucocorticoids and mineralocorticoids and androgens, all three will be decreased if you have primary adrenal insufficiency. If you have secondary adrenal insufficiency, then you decrease your ACTH, you decrease your glucocorticoids and your androgens, but your mineralocorticoids stay the same because ACTH does not affect mineralocorticoids or aldosterone. So pigmentation, so we said that excess ACTH causes pigmentation. Why is that? So it's because ACTH is derived from proteolytic processing of this molecule called PMOC, which is pro-opiomelanocorticoid protein. So this is the N terminus, this is C terminus, and you can see that processing of this leads to ACTH production, but it also leads to production of other hormones such as MSH, melanocyte stimulating hormone, and increasing the POMC processing causes increased MSH, and that causes increased pigmentation. So it's a side effect of increased ACTH. Let's have a closer look at cortisol, which was the stress hormone, the glucocorticoid secreted by the adrenal gland, the zona fasciculata, and it does these things. So some of these will be expected. So it has these six main roles. So firstly in starvation, that's probably its most important role. Then in inflammation, the immune system, fetal development, the central nervous system, and the cardiovascular system. So it's a diabetogenic hormone, which means that it increases blood glucose and acts in synergistically with insulin, and it clashes with it acts antagonistically to insulin, and it acts synergistically to glucagon and that sort of thing. So starvation, cortisol will increase the blood glucose, so the body can use it for fuel and protect the liver glycogen reserves. In inflammation, so what happens in inflammation is that an infection causes immune cell proliferation, and then this releases some chemicals such as nitrogen oxide, prostaglandins, and leukotrienes, which cause inflammation. So cortisol inhibits the synthesis of these three and other inflammatory substances, which decreases inflammation. In the immune system, we have when macrophages devour the pathogens, they secrete interleukins, and these interleukins potentiate the hypothalamus and pituitary activity, which then will obviously lead to an increase in cortisol. But then cortisol itself decreases the secretion of interleukins from macrophages, so it keeps itself in check and inhibits its own release during infection. Now, in fetal development, late in gestation, the fetal adrenal gland secretes cortisol in preparation for birth. So cortisol has an important role in birth and fetal development. It causes surfactant production, which is important for lung recoil, liver glycogen deposition, so the fetus, so the baby, when it's born, it has glycogen reserves already in case food isn't readily available. It causes digestive enzyme secretion and stomach acid secretion as well, so it's important for digestion, increases kidney filtration, and causes T4 to be converted to T3. We'll look at the thyroid hormones later, but that's important for growth and metabolism. So it also has a role in the central nervous system. It can cross the blood-brain barrier and alter the mood. For example, in jet lag, it's thought that our cortisol levels are not in sync with real time, so that's what causes jet lag. Now, cardiovascular system. So cortisol permits catecholamines to increase vasoconstriction for the fight or flight response. It maintains blood pressure and increases EPO, erythropoietin, which increases blood cell production, red blood cell production, which has a role in maintaining blood pressure as well. So lastly, we're having a look at the catecholamines and some disorders. So the catecholamines are secreted from the deepest layer, the adrenal medulla, which is part of the sympathetic nervous system, and chromatin cells secrete these catecholamines, which include adrenaline and noradrenaline, or epinephrine and norepinephrine if you're in the US. So it secretes catecholamines under stress, which causes the flight or fright response, increases heart rate, increases cardiac output and contractility of the heart, increases systolic pressure and decreases diastolic pressure by dilating vessels in some areas, such as muscles, and constricting elsewhere, to divert the blood into the most appropriate place. Causes piloerection, so hair stand on end, and pupil dilation, so more light can get into your eyes, I think. Decreases gut motility, because you don't need your gut when you're needing to fight or fright. And increases glycogenolysis in the liver. So how is it made? We had a look at how cortisol is made. Let's have a look at how catecholamines are made. So they are amine hormones derived from tyrosine. Tyrosine converted to dopa, which converted to dopamine, noradrenaline, and then adrenaline. So the key enzymes here are tyrosine to dopa, we use tyrosine hydroxylase, dopa to dopamine, we use dopa decarboxylase, dopamine to noradrenaline, we use dopamine beta hydroxylase, and noradrenaline to adrenaline, we use phenylethylamine and methyltransferase. Yeah, so the last bit of this is adrenal endocrine disorders. So we can have various endocrine disorders. So we'll start with hyperadrenocorticism, which is an increased level of cortisol in the blood. So this can result from an adrenal tumour, increased ACTH levels, increased corticotropin releasing hormone levels, increased corticosteroid treatment. It has symptoms which are similar to diabetes, so this is known as Cushing's disease. We have things like weight gain, rosy cheeks, skin pigmentation, skeletal muscle wasting, purple abdominal striations, capillary fragility, immunosuppression, and glucose intolerance. Now, we can have these various hypoadrenocorticism diseases, so not enough cortisol, and they can be primary or secondary, as we looked at before. So primary hypoadrenocorticism, or Addison's disease, is caused by deficiency in glucocorticoids and mineralocorticoids. It's caused by destruction of the adrenals and autoimmune disease and hemorrhage. So we have a lack of aldosterone and cortisol. We have tiredness and weakness, vomiting, skin pigmentation, hypotension, and death. Secondary hypoadrenocorticism is not caused by destruction of the adrenals, but deficiency in glucocorticoids only, caused by lack of ACTH. This leads to cortisol and sex hormone production impaired, loss of body hair in females, but not in males, because males get sex hormones from the testes, but surely it has other effects too. So now we look at congenital adrenal hyperplasia, which is caused by lack of enzyme involved in cortisol production. It's most likely the 21-hydroxylase deficiency. And this actually has the opposite effect, so it causes hyperplasia by increasing the CRH and ACT levels to compensate for this lack of cortisol. And this causes excessive sex steroid production as well, which causes masculinization of the female fetus in utero. This can cause clitoral hypertrophy, fusion of the labia and urogenital opening at the base of the pseudophallus, and is often treated with surgical reconstruction of the genitalia, because it's usually purely a cosmetic thing. Now let's have a look at thyroid hormones. So this is what the thyroid sort of looks like. It's underneath the thyroid cartilage. It's attached to the trachea below the larynx, and it has two lobes connected by an isthmus. The parathyroid glands, 4 of them, are behind there as well. It has autonomic innervation and secretes these 3 hormones: T3, triiodothyronine, T4, thyroxine, which are amine hormones, I think, and both contain iodine. And then it also secretes calcitonin, which regulates serum calcium levels. So if we have a closer look at the histology, it's made of many of these colloids, and the colloid material contains T3 and T4 bound to thyroglobulin. It has lymphatic vessels, good lymph drainage, and synthetic nerve endings. There are C cells surrounding it, which are otherwise known as parafollicular cells, and these secrete calcitonin. Calcitonin regulates plasma calcium concentrations, has a basement membrane and epithelial cells surrounding the colloid. The sympathetic nerves control the thyroid blood supply. So the thyroid HPA axis then. So we had a look at the adrenal HPA axis. This is just the same thing, but with the target organ being the thyroid gland organs. So the hypothalamus, it releases thyroid thyrotropin releasing hormone. to the anterior pituitary, where a thyrotroph will release TSH, thyroid stimulating hormone, and TSH is important for thyroid hormone synthesis inside the thyroid cells. So inside these colloid cells, this is where T3 and T4 are synthesized. So the steps of that which require TSH are iodine uptake, oxidation, thyroglobulin, endocytosis, and T3 or T4 secretion, which we'll have a look at the whole process later, but that's just for now. Thyroid hormone synthesis involves tyrosine, which gets converted to monoiodotyrosine and diiodotyrosine. Monoiodotyrosine gives rise to T3 and diiodotyrosine gives rise to T4. T3 is the most potent version, so T4 is converted to T3. T4 can be converted to RT3 as well, which is a reverse T3, which is inactive, so not that useful. And then the T3 and T4 travel to the target organ. T4 is converted to T3 in the target organ as well via a deiodinase enzyme, which has negative feedback on the hypothalamus and the anterior pituitary, so a long indirect loop and a long direct loop as well. T3 has many cellular and whole system effects, so it binds to an intracellular receptor. So it's a lipid hormone. It involves transcription and protein synthesis and increases cellular respiration and growth, which increases food intake, increases the basal metabolic rate and oxygen consumption, and increases cardiac output and ventilation, as well as thermogenesis as well. It has important effects on growth, maturation, skeletal nervous system, and mental development. So let's have a look at thyroid hormone synthesis, which was this step here. So we have the blood and the colloid. So firstly, we need to uptake of iodide ions, which it happens via a SLC5A5 symporter, which is a sodium iodide symporter. So swap sodium ions for iodide ions. I've only drawn the iodide ions here. So the iodide ions enter, and then two of them are oxidized to produce electrons and iodine. This is potentiated by TSH, so TSH is important for uptake and oxidation. Iodine exits the follicular cells into the colloid space from the blood, where there's thyroglobulin with tyrosine residues. So the iodine then iodinates some of these tyrosine residues, so forming diiodotyrosine and monoiodotyrosines on the thyroglobulins, and then coupling between the iodotyrosines causes formation of T3 and T4. These thyroglobulins are then endocytosed into the follicular cells, and you can see these vesicles carrying them, which fuse with lysosomes, and the lysosomes break down the thyroglobulin and T3, T4, and then release the T3 and T4 into the blood, so secretion, which TSH is important for secretion and endocytosis of thyroglobulin as well, so it has many important roles in thyroid hormone synthesis. Now, thermogenesis is an important effect of thyroid hormones. So thermogenesis was one of the effects we looked at. How does this happen? So there's two types. There's peripheral thermogenesis and central thermogenesis. Peripheral thermogenesis is T3 having an effect on the muscle, whereas the central is T3 having an effect on the ventromedial hypothalamus. So T3 affects the muscle, which activates these things. So sodium potassium ATPases, GPDH, and uncoupling protein UCP3. And in the endoplasmic reticulum, calcium is released via SERCA, which causes heat. So this SERCA is an ATPase, and all of this together leads to thermogenesis. Central thermogenesis is T3 affecting the ventromedial hypothalamic neurons, and then the VMH causes decreased AMPK and decreased sympathetic nerve activity, which causes brown adipose tissue to release heat via uncoupling protein 1. So let's have a look at some thyroid disorders. So we have hypothyroidism and hyperthyroidism, too low, too high. Iodine deficiency, decreased thyroid stimulating hormone from the pituitary, thyroid disease, and lack of thyroid can cause hypothyroidism, which leads to weight gain, agoitre, which is the enlargement of the thyroid gland, cold extremities, lethargic, muscle weakness, decreased pulse and cardiac output, cretinism, which is a form of severe mental retardation if there's very severe hypothyroidism during fetal development. So these are basically decreasing your metabolism, and those are the effects. Thyroid overactivity can be caused by thyroid hyperplasia, thyroid adenoma, and 50% of autoimmune diseases can cause this, so Graves' disease, for example. This will cause hypothyroidism and cause the opposite effects, most of these, but there are some common effects like weight loss. Agoitre is possible, sweating, agitated, nervous, muscle weakness, increased pulse, muscle weakness because the amino acids are broken down. That's an effect of cortisol. On metabolism, increased pulse, atrial fibrillation, increased growth and bone maturation, staring eyes, and because this is an immuno disease, autoantibodies can cause a muscle to swell and push the eyes forward, which causes these staring eyes. And then you can have infertility and lack of periods in women. So treatment of thyroid conditions, we can use block and replacement therapies, we can use surgery and radioiodine treatment as well. So having a look at growth hormones now. So firstly, what is growth controlled by and what are the normal patterns in humans? So growth patterns in humans, growth is fastest at 16 to 20 weeks of gestation in development. So this is all about development. And the earlier growth spurt, the shorter the final height, which partially explains why girls are usually shorter than boys on average. Well, women are shorter than men when they grow up. So we can have a look at this graph, which shows the individual organ growth. So not all organs develop at the same rate. Most of them finish developing to 100% size at 20 years. But we can see that the reproductive develops later. General is in the middle, and then the brain and head develops earlier. Lymphoid organs develop in the middle, around 10 to 12 years. So there's three main factors involved in growth. So what affects and determines growth? We have genetic factors, environmental factors, mainly nutrition, and physiological factors, which are mainly growth hormone and the other chemicals that we looked at. Now, the structure of growth hormone, which is the principal hormone controlling growth in humans, is known as somatotrophin, and it's a 191 amino acid polypeptide. Has a tertiary structure and two internal disulfide bridges. It's related closely to prolactin and placental lactogens in its structure, I think. So let's look at the HPA axis for the control of GH. So the hypothalamus can release growth hormone releasing hormone, or somatostatin. So growth hormone releasing hormone is a 44 amino acid peptide hormone, and it's released from the arcuate neurons, which affect the somatotropes in the anterior pituitary. It signals via GS signaling. The hypothalamus is also secreting an inhibitory hormone called somatostatin from the periventricular neurons, which decreases the activity of the anterior pituitary with respect to secretion of growth hormone. Now, the anterior pituitary secretes growth hormone, which itself has a short loop negative feedback to the hypothalamus. It's diabetogenic, so it increases blood glucose, synergistic with cortisol and glucagon, but antagonistic to insulin. Now, the growth hormone has these primary effects and secondary effects, so we can have a look at the primary effects on the liver, muscle, and adipose. So on the liver, growth hormone increases protein RNA synthesis, increases gluconeogenesis and glucose utilization. On the muscle, it increases amino acid uptake and protein synthesis and decreases glucose uptake. On adipose tissue, it increases lipolysis, increases free fatty acids, and decreases glucose uptake. So all involved in growth and increasing the size of cells, using more energy. Other effects include increasing sodium and fluid retention, increasing glomerular filtration rate, which causes these things in the kidneys, increasing the basal metabolic rate, and decreasing insulin sensitivity, because it's antagonistic to insulin. Now, the secondary effects are mediated by the liver. So growth hormone, one of the effects of growth hormone on the liver is to cause it to increase release of IGF-1, which is a somatomedin. So IGF-1 has the secondary effects, which are, it has effects on the liver, increases growth more, increases DNA synthesis, and increases collagen production. On the viscera, so other organs, it increases protein synthesis and increases the cells in size, so clearly growth there. One of the most important effects it has, secondary effects, is on bone ossification. So bone starts as a cartilage model. There's primary ossification in the shaft and then secondary ossification in the epiphyses. And IGF-1 causes increase of chondrocytes size, continued growth at the epiphyseal growth plate. On soft tissue, it also increases growth as well. And these are the long feedback loops. So IGF-1 produced by the liver has an indirect long feedback to the hypothalamus and a direct feedback to the anterior pituitary. So these are some other factors affecting GH release. So obviously growth hormone releasing hormone, that increases GH release. Arginine, ghrelin increases GH release. Hypoglycemia, low blood sugar, because it's antagonistic to insulin, and glucagon would be released when there's low blood sugar. So it does the same thing. And sleep, exercise both increase growth hormone release, because growth hormone is released more during the night when you're sleeping. Factors decreasing growth hormone release includes somatostatin, obviously, and then we have growth hormone itself, if too much is produced, and IGF-1 via negative feedback. Hyperglycemia, because it's synergistic with glucagon and antagonistic to insulin. Increased age, thyroid deficiency, and lipids. Other hormones controlling growth are growth factors, thyroid hormones, gonadal hormones, glucocorticoids, and insulin. So growth factors, these are small secreted peptides. They don't travel all the way in the blood to the rest of the body, but they are more local and specific. Some examples are transforming growth factor, fibroblast growth factor, platelet-derived growth factor, erythropoietin, increasing the number of blood cells, which is a growth factor 2, and interleukins, which stimulates the growth of immune-related cells. Thyroid hormones cause maturation of the central nervous system, and hypothyroidism causes short stature as well. Gonadal hormones, so we have systemic effects of gonadal hormones and local effects. Systemic effects include increasing growth hormone to increase it for the pubertal growth spurt, and increasing the rate of long bone growth. The local effects include sex steroids, which trigger epiphyseal fusion and limit long bone growth, actually. Now, for glucocorticoids, for example, cortisol, if they're present in excess, they can decrease growth. And number 5 is insulin. Growth hormone disorders. So growth hormone excess can cause these disorders called acromegaly and gigantism. So acromegaly is increased growth hormone after puberty, whereas gigantism is increased growth hormone before puberty. Gigantism tends to be more proportional, whereas acromegaly affects the face and it can look a bit different from other people. So acromegaly is quite rare, it's about 60 per million. It's often caused by pituitary tumour and common in cats as well. Increased cartilaginous growth results from it, so large ears, jaw, nose, hands, feet, and tongue where there's cartilage. Treatment involves surgery, radiotherapy, dopamine agonists, and somatostatin analogues, which can all decrease the growth hormone secretion and activity. These are for treating a pituitary adenoma, which can cause increase of growth hormone secretion. Growth hormone receptor antagonists can help as well, so it doesn't have that much of an effect. If we have GH deficiency, then that can cause pituitary dwarfism. So decreased growth hormone in childhood means decreased growth, and pituitary dwarfism results in normal body proportions, however. You can have treatment, which is injection of a species-specific growth hormone. Pituitary dwarfism is not to be confused with achondroplasia, which is unrelated and causes abnormal body proportions and is caused by a mutation in FGFR3 gene, and this also causes dwarfism but with abnormal body proportions and is not related to growth hormone. So the last thing I'm going to have a look at is hormones involved in energy balance. So let's get acquainted with the units of energy used first. So energy is measured in joules normally. This is the work done when 1 Newton moves 1 meter. Power is the rate of work, so 1 watt is 1 joule per second, or newtons per meters per second. A kilocalorie is the heat required to warm 1 kilogram of water by 1 degree Celsius, and 1 calorie is 1,000th of that. So the heat required to warm 1 gram of water by 1 degree Celsius. 1 kilocalorie equals 4,184 joules, if anybody was interested in that conversion. Now let's have a look at what basal metabolic rate is. So BMR, or basal metabolic rate, is the energy required to keep a resting awake individual in a thermal neutral environment, and that's about 1,800 kilocalories per day for an average person. It's measured by indirect calorimetry, by measuring the amount of heat generated in oxidation by determining the intake or consumption of O2, or by measuring the amount of CO2 released. So these are some approximate formulas. The metabolic rate in kilocalories per hour equals 4.8 times the VO2, so the consumption of oxygen, or 3.9 times the VO2 plus 1.1 times the release of carbon dioxide, VCO2. Things that increase BMR include infection, high temperature, increased growth rate, pregnancy, lactation, menstruation, large size, stress, and being male. Decrease BMR are related to low temperature, old age, starvation, and being female. So endocrine control of energy storage goes like this. So insulin and glucagon are the principal hormones involved. We also have some incretins, and other things are regulating appetite, as we'll see later. But first of all, blood glucose. So homeostasis of blood glucose is important. It's around 70 to 110 milligrams per deciliter. That's the average normal glucose range. So when glucose increases in the blood, the beta cells in the islets of Langerhans of the pancreas secrete insulin and increase the energy storage, so they suck the excess glucose away from the blood and into the tissues. Insulin is potentiated by incretins, and incretins also decrease glucagon release. The incretins are gut peptides such as GLP-1, glucagon-like peptide, GIP-1, and oxyntomodulin, OXM. This causes the rise in blood glucose to be reversed, and by increasing glucose uptake and utilization, increasing glycogenesis, so glucose conversion to glycogen in the liver, and muscle, increased protein synthesis and increased synthesis of triacylglycerides. And the opposite happens in decrease of blood glucose, so the alpha cells in the islets of Langerhans of the pancreas secrete glucagon, which decrease energy storage, so it's like shoots out all the glucose. So glycogen is converted to glucose, increases fat conversion to fatty acids, and increases synthesis and release of glucose as well. And that increases the blood glucose concentration to return it back to normal. Now, regulation of food intake by the hypothalamus. So anorectic hormone is an appetite suppressant, whereas orectic hormone stimulates the appetite. So this is a view of the brain, the hypothalamus, and this is the lateral ventricle, cerebral cortex, and the third ventricle. So you have these two satiety centers here, these ventromedial nuclei. These control satiety, and if you destruct them, then you can cause hyperphagia and obesity because they're no longer sending out the factors that tell us that we are full and don't need to eat anymore. The lateral hypothalamus nuclei control the feeding center, which tell us that we're hungry and we need to eat. If you destruct these lateral parts, then you decrease food intake. Leptin is a satiety hormone. It's a 16 kilodalton protein produced by the white and brown adipose tissue. So the fat cells in your body secrete a satiety hormone, a protein which travels in the blood to tell you that you're full. Leptin in the blood is proportional to the amount of adipose tissue in the body, so the more fat you have, the more satiety you're meant to feel, apparently, according to this hormone. And mutations in the leptin receptor are found in some obese humans, so the body is not responding to leptin, so that's why they don't feel hungry. They don't feel full when they also have a lot of fat in their body. Leptin crosses the blood-brain barrier to reach the hypothalamus and regulate feeding behavior. So we have experiments which show that this is true. We can use mice with leptin gene mutation, called OB mice, which are obese, or mice without a leptin gene receptor mutation with diabetes, so DB. These two types of mice with impaired leptin, either leptin secretion or leptin reception, have these symptoms. So they're hyperphagic, they're obese, have a lot of fat in their bodies, and increased type 2 diabetes risk. If you inject leptin into an obese mouse with a leptin gene mutation that can't produce leptin on itself, then it can produce weight loss. But if you do the same, if you inject leptin into a diabetes mouse with a receptor gene mutation, then no matter how much leptin you add, it doesn't have the receptors to respond to it. So it also contains higher than normal leptin levels. So adding more isn't going to help at all, and it's not going to lose weight. These leptin mouse experiments show that this is true. So parabiosis is when the obese rat generated by a ventromedial lesion had blood supply connected to a normal rat, which is a parabiotic rat pair. DB attached to, so you have to imagine these are mice, but a diabetic mouse attached to a lean mouse. Diabetic mouse produces high leptin, but it's not responding to it itself, but then it affects the lean mouse. The lean mouse has its feeding suppressed and it dies of hunger. Well, not of hunger, but just no food, because it's too high leptin levels. And then DB connected to OV also causes weight in the obese mouse because of the high leptin. However, an obese mouse connected to a lean mouse is not producing enough leptin, so there's going to be no change. And lean mouse to lean mouse, there should be no change as well. There might be some decrease in the fat pad size. Now, ghrelin is one of the only hormones, there are very few, that increase food intake. So it's a 28 amino acid peptide produced by PD1 cells in the stomach fundus and the epsilon cells in the islets of Langerhans. It's orectic and decreased after a meal. It stimulates neuropeptide Y and agouti-related peptide neurons in the hypothalamus, and increases growth hormone secretion from the pituitary as well. So remember, that was ghrelin was one of the factors that increased growth hormone secretion. Now we have a look at how these hormones interact in the brain to regulate food intake. So the important areas are the arcuate nucleus, the hypothalamus with second order neurons, and the effectors that it affects. So we have adiposity signals, satiety peptides, and hunger signal ghrelin. There are two main adiposity signals released from fat, so leptin and insulin. Leptin increases the POMC or CART, it attaches to those receptors, which stimulates alpha melanocyte stimulating hormone, MSH, which is a anorectic peptide, and stimulates these MC3 or 4R second order neurons in the hypothalamus, which causes decreased consumption and increased metabolism. Satiety peptides include, and insulin also inhibits neuropeptide Y and agouti-related peptide released from the arcuate nucleus. So satiety peptides include PYY, PP, pancreatic peptide, GLP-1, and oxyntomodulin, OXM. So these satiety peptides released from the gut inhibit the NPY expression, which itself inhibits POMC and CART, and agouti-related peptide also inhibits the MC3-4R in the hypothalamus. The MPY and the agouti-related peptide increase the Y1 and Y5R in the hypothalamus neurons, which increases consumption and metabolism. Hunger signal, ghrelin, is one of the few hormones which are orectic, and it positively affects MPY and agouti-related peptide, which positively affects Y1 and Y5R, which increases consumption and decreases metabolism. And there are also vagal afferents, so the amount of stretch of the stomach and the gastrointestinal tract that information is sent via the vagus nerves to the nucleus tractus solaris in the brain, and that can communicate with the hypothalamus as well. In the hindbrain, this information is processed and can cause changes to feeding, gastric emptying, and metabolic rate. These are a summary of the signals regulating food intake, so most of these are anorectic except for ghrelin. Leptin, insulin, and ghrelin, these are peripheral signals sent about energy stores, often from the fat, and nutritional status. Neuronal pathways, for example, the vagus nerve, which affects the DVC, so info about the stomach distension and food digestion can be sent via these vagal pathways and cause a change in feeding behaviour. CCK, cholecystokinin, is released via the vagus nerve and stimulates satiety. Oxyntomodulin, PPY, and GLP-1 decrease food intake and are released from the lower gastrointestinal tract. PP, or pancreatic polypeptide, is released from the islets of Langerhans and decreases food consumption as well. So what happens when this system goes wrong? So treatments for obesity, we can just have caloric restriction, which might be difficult if the receptors, if the leptin receptors are not being transcribed properly. Caloric restriction or bariatric surgery, which these are 3 types of bariatric surgery. So we can have vertical banded gastroplasty, adjustable gastric banding, or Roux-en-Y bypass, the most aggressive obviously. Gastric band just creates a diversion or creates a small stomach pouch, which you can inject saline through a port to increase the tightness or decrease the tightness by sucking some out. And food collects here before going through the rest of the stomach, which makes you feel full faster. A Roux-en-Y bypass, which connects the jejunum to the esophagus, and it bypasses all of this, so you feel full faster, and it's malabsorptive as well, and decreases ghrelin levels. Apart from caloric restriction, you can also do exercise, have liposuction, which is more cosmetic, and restrict food absorption with a drug called Xenical, which is a lipase inhibitor and prevents lipid digestion, but the side effect is steatorrhea, which is too much fat in your stools. So that's an overview of the major systems in the endocrine system, and also the principles as well. Hope you enjoyed this video. Give it a thumbs up and subscribe if you like this video, and thank you very much.